Hi, everybody. So I spoke yesterday about how the idea of a CACNA1A, CACNA1A database came about. Well, Dennis Lal is the one, there he is, Dennis Lal is the one who made that idea a reality. In addition to the portal, Dr. Lal is here with his research team. They have put together posters discussing their work, which are displayed in the hall. So I think what we'll do is during lunch, Alina, Tobias, and Arthur will be standing outside, and please feel free to go up to their posters and ask them questions. They'd love to discuss it with you. The aim of this portal is to enable global aggregation of clinical, genetic, and molecular data for CACNA1A variants. It's still a work in process as scientists begin to submit their data. I'm excited to introduce Dr. Dennis Lal from the Cleveland Clinic, who is here to unveil the CACNA1A portal.
Thank you very much. Okay. Yeah, I'm really happy to be here. As it turns out, our group is working really a lot on ion channels in the last years, and I think we have learned a lot of lessons which are transferable for CACNA1A. And the whole point I want to make with this presentation is we're living now in a time, where it's good to live in and to be hopeful when we think about rare disease, because basically the technologies are there. They just need to be individually adapted for every gene to develop new types of therapies. And if you just observe a little bit, if you type in, for example, genetic therapy, targeted therapy, biotech, and you type in, for example, the words developmental disorder or epilepsy or autism in Google, you will see that there is craziness going on. So it's really a good time where a lot of companies have been built. So if you say these days you are a biologist and you are studying, for example, an ion channel, your career today is not necessarily anymore that you become a weird professor, like I am, for example. So it's actually these days it's really hard to recruit people to stay after they have their doctorate in a research lab, because most people go to companies now. It's very different, like even 5 years ago. So suddenly biologists have real jobs. They can develop drugs. Before it was more like for the chemists. So, but we're living in a really different time now. So, and the beautiful thing, what we have learned for the ion channel disorders is that we have now a game plan. It's not anymore random. It's a path from figuring out that there is the gene, there is a mutation, towards how can we develop drugs which can, tackle that problem, is pretty clear these days. However, it requires certain homework which needs to be done, that we are in the situation that big companies feel comfortable to do the investment, to adapt existing technologies. It's important, existing technology, that it works for CACNA1A. So, and the whole reason why we are here is to remove these things which give a little bit uncertainty. So do the homework that big companies feel comfortable investing in drugs or repurposing frameworks which have been developed for other ion channels over the last years. Okay, so we are very close, but we need to do a little bit more homework as a community. Okay, so I will talk just very briefly about what our lab is doing, and then I will go a little bit about the bigger picture why I'm here. And if you want to see a little bit more about the type of research we are doing, we have like 3 posters from Alina, Tobias, and Arthur, who are sitting there, and they have basically done, together with Eduardo, most of the work I will present today. And feel free to talk with them, bring them some food. They are not allowed to eat during the lunch break until they have talked to every one of you. And so that will be good. Okay, so the interesting thing is, these kind of flow charts or like plots you see quite a bit, is that there's a game plan now how to develop drugs, and with this biotech type of way. And so what is typically done is like you identify like a target, that's a fancy word for most of the time gene, and then you do some, you execute the game plan as a biotech company, you find ways to develop a drug or a molecule or whatever, and to change, how that, gene or protein at the end works. And there are many, many ways how you can do this in a very fast and efficient way. And then you refine it so that it's really specific, that you don't get surprising results once you have found it. And then you test it in animal models because you don't want to go directly to humans. And then you can do a little bit more things to, make the drug work, and that you feel very comfortable that you can say, hey, FDA, let's go into humans because we think we are ready. So, and this upper part is where billions of dollars went now the last 5 years in particular. So this is really where now the basic research, which was funded by governments and foundations, is now taken on further by companies who want for-profit develop, therapies. And this is where a lot of energy is spent, and there's a lot of money in that area. However, what happens now more and more frequently, and this is what we observe for more advanced disorders, where this part has basically been done, is that there are no drugs. However, it's not really clear which version of the disease would benefit from that drug. Because what we have learned, particularly from the ion channels, it's not one gene, one disorder. And everyone in this room knows this, right? If we just look around, we see there's complexity. So it turns out that one gene can cause disease in different ways, depending on where and what is modified. So and frequently what happens is that you have one or two drugs, but you don't know really who in this room should benefit from drug A or from drug B. And this is a big problem. Because typically when a pharmaceutical company has reached that stage that they have a molecule which they think is ready for clinical trials, at that point they have maybe 1 or 2 years money to execute the trial and show enough results, because it's the ultra expensive phase, to show success and survive longer. However, here this might be a significant roadblock if you don't know where all the patients are and who should benefit from this. So that's why it's really important to understand where are all individuals who have CACNA1A. So which hospitals are they, where do they live, how can we contact them, and who are their treating physicians. And also, who would benefit from a drug which might work for patient version A, and who are the patients who would benefit from drug version B. And this is work where there's not a lot of money. So, and this is then where short notice or like last minute type of solutions are trying to be found, and which often don't work. So here's really where our lab is focusing on, and with a lot of collaborations here in this room, where we try to figure out so-called genotype-phenotype correlation, what mutation leads to different type of version of the disease, how can we make sure that a variant is really causing disease, and what is the underlying mechanism, and here's a lot of collaborative work been done. And then since there are many, many different mutations, maybe we can learn from some mutations which have been very well studied, and can learn and take this knowledge and move it to other disorders. So a lot of this research is basically this lower part has also a game plan, which is currently very defragmented. And we, now in this presentation, I will try to make the point that we need to bring everything together. So, and a lot of these things is really, associated with also education. So, a good trick is to see if you're in good hands, if you are going to your care provider, and you ask your care provider, does my son or my daughter have a gain or a loss of function mutation? And if this question, you cannot, in general, you cannot expect an answer, right? But if your caregiver is not able to understand the question, then you are not in good hands. That's really important. So there's some things, treatments where you don't need to, but not everyone needs to be a specialist in genetics and the mechanism for everything. But if you really want to go one step further and you have one main caregiver which is working with you, that person should at least have heard the words gain of function, loss of function. And if that's not the case, go to a different doctor. And I can say this, I'm a biologist, so nothing what I say is meaningful. So now moving on now to CACNA1A. So one thing is very interesting, and I hope that it's giving a lot of people hope here. So when we listen to the previous talks and also the foundation, so this community is a really relatively young group, right? So the foundation was just recently been founded, and there are in our databases only a few hundred individuals now. And this is really surprising if you look a little bit at the history. So these are genes which are associated with neurodevelopmental disorders and in which year they have been for the first time associated, in this case, with seizures. And you see, on the one hand, why medical doctors are really struggling giving good care for every gene, because every gene is different and you see how many are there, and they're coming more and more. And if you have like a 50-hour week, how do you catch up, right? It's almost impossible. And even for genetic counselors who can be specialized on neurological disease, it's also hard, right? So, and the interesting thing is, if you look for when CACNA1A was first time associated with seizures in that case, and migraine is even earlier, it's quite some time, right? So there is, this is not a disease which has been associated, it's not a gene disorder which has been associated with disease just yesterday. There's a lot of history within this gene. And the interesting thing is, if you, for example, think about disorders of the brain, which typically arise in childhood, you can also look at what types of genes and, at the end, this molecule protein are these genes. And you can see, hey, the main group are ion channels. And CACNA1A is one of them. So, again, I will come more and more over to this point that we can learn from genes, disorders which are already in clinical trials, right? So we don't need to reinvent everything from the beginning. So CACNA1A is an ion channel, right? So like many other ion channels, which are monogenic, which have complex, disorders where not every patient looks the same. And there are also game plans in place to move still to clinical trials. So the interesting thing is, if you look at this slide here, so the size of each square is based on the number of pathogenic classified variants, which have been reported in the literature. So you might think, CACNA1A is maybe not such a big player in the game, right? It's an ion channel, it's relatively, among many other genes, it's relatively big, but it's not such a big player. And I would argue that CACNA1A is probably the most frequent neurodevelopmental disorder. So the most. And as a geneticist, I feel relatively comfortable talking about this because at the end, when it comes to number of patients, it's very strongly correlated with the size of the gene. And CACNA1A is among all these ion channels which you observe here, the largest. And the only other factor which leads to the number of patients is how many times a mutation can be passed on. And this is typically associated with milder forms of the disease. And among all these neurodevelopmental disorders here, CACNA1A has a relatively large fraction of individuals who have a relatively mild phenotype. That means we have more mutations in the population, and that means that this disorder will become the most frequent one. That is very clear right from today. So, what is the problem? So, when we now compare, for example, to CACNA1A to other, ion channel disorders, wherein we look at in a database where geneticists and genetic counselors enter their mutation after they have interpreted that variant. So in the US, for example, if you are a genetic counselor and you look at a clinical genetic test, you then upload your interpretation of a variant which you found in an individual, in a patient. And what you observe here is that we have tons of individuals who have variants, but they're in this category, which was yesterday also mentioned by the CHOP team and GeneDx, that these variants are so-called variants of uncertain significance. So the point is the variants, so we have many patients or potential patients, but we are not really confident enough to say that is CACNA1A. Because the situation often is, if you do a genetic test, that you have multiple genes which have a rare mutation or variant, and which in theory, all these genes could explain this neural developmental disorder. So, and we are not in the position at the moment to say that patient, your child, looks exactly how CACNA1A children version 2 look like. We're not in the position. This is where the homework we need to do. We need to better understand among, for example, migraine, this is the exact critical feature of this version of migraine, which is only seen in CACNA1A. And we only get to that level when we have really well groups of individuals who have really state-of-the-art, cutting-edge assessments, clinical assessments, and we can compare everyone who has, for example, CACNA1A and migraine compared to people who have migraine but not CACNA1A. And then things become very clear. And then we can have rules, and then we can write these rules down, and then it becomes also clear for everyone in the world what is the typical version of migraine or ataxia or epilepsy associated with the gene. And then this big bar here, this VUS bar, will become smaller. Okay. So this is the challenge, right? So removing this VUS bar, and again, reoccurring again, this is a problem which we not face only for CACNA1A. If you look at across all these disorders, this is the #1 challenge. And this is the challenge to make a diagnosis for the clinician at the end with the help of the genetics team, if an existing therapy should be started based on the genetic test or not. So if it's a VUS, you should not make a clinical intervention based on that gene because you're not certain that it's pathogenic. So at this point, this is a problem which is not great, but it's not life-threatening often. So because we don't have perfect drugs yet. However, this will change in the next few years dramatically. So this is a problem which is really a huge one, and there's even a bigger problem. I come to this in a second. So to once again bring this point home, because not everyone, so actually it's, even in the scientific community, this is not much appreciated. So if I would remove the labels and I would ask Al and Jen and other molecular experts, which one is CACNA1A, which one is SCN2A, which is SCN1A, and I give them 5 seconds chance, probably that's not so easy to distinguish between these ion channels. And this is because they are evolutionary very similar. So I always like to make this example. If you consider this as an essential building block which does something in a human cell, let's call it a neuron, so a brain cell, this could be also like an essential part of a car. Let's call it like an engine, okay? And an engine in a Volkswagen, I'm a German, right? So Audi or Mercedes, all German cars, are still engines, right? So if you don't see the rest of the car, you might not be able to, and don't see the logo, you might not be able to distinguish them. At the end, they have all engine problems, and different parts of the engine problems lead to different kind of effects. Car may go faster if you, I don't know, have specific, I'm not a mechanic, okay, but you get the point, right? So typically they go slower. So, and the interesting thing is also, if you now look where at these parts of this protein, so that's a fancy word for this molecule, where would you have the most patient variants, and where are also the regions which are most evolutionary conserved. In biology, we think if something was already there with, a couple million years back, then it must be more important than something which just came a few years back and had some function. And when we look at these regions, which seem to be important, they seem to be important in all of them in similar regions. So again, making the point that we don't need to start every time from scratch. They are not identical, I don't want to say that, right? But there are certain lessons which we can learn from other channels. And this can boost and de-risk certain information and step forward just using this. So why is this whole interpretation important? So, and this is really, close to my heart. So, when we have a classical drug, we have a severe disease and we don't, and we want to see and we want to help someone, the situation is the following. You give the drug, and if you don't, and we have this so-called, here's the drug concentration, and then we have this so-called therapeutic window. Imagine you have a drug and you need 100 points that the drug is working properly. If you only give 80 points, that drug has not, it's not working because you don't have 100 points. If you give 200 points, that might be too much drug, that might be bad, right? So, but if you are giving, let's say, 100 points or 110 points, you are in this therapeutic window, and then the drug is working perfectly. So this is where you want to go. And, with different, people, different weights, different amount of blood, different amount of foods, whatever, it's not so easy to hit this, therapeutic window. So, but in the classical drug, there might be a range, and you can try and error, because if you maybe shoot one time a little bit too high and you see some adverse effects, you just wait, it will, because the drug will be cleared out of the system at some point. So you can, try and error a little bit. So, and imagine you have a disease where you have that your neurons are too excited, so they are always firing, and that's why you have, for example, seizures. You can imagine, okay, you give the drugs not enough, and you still have seizures. You cannot push it over here, but then you give more, and suddenly you don't see any more seizures. Things are good, right? So this is how we think about classical drugs with classical disease. However, what you have noticed with, what also in Jen's talk and the talk before that, what has been reported is that we have a more complex situation here. So we don't only have one disease, we have different diseases, and which are somehow also related. And how we think as biologists about this is that there's a gradient. So there's, if you have too much CACNA1A, that's not good. If you have too less, also not good. So you want to be, that your body is within this range. So then, and if you, for example, would think that you have a drug which is too bad and you give that over a long time, you might get opposing effects. And this is not really understood. And we don't have good measurements at the moment how to understand if we are within the therapeutic window, so let's say here, or if we are already here. Because these effects might not turn out to be there the next day. They might develop over time. So this is something where we as a clinical community needs to have also really high resolution data to monitor these kind of effects. And this is only possible if we bring the data which we have in one center with a patient with a specific mutation together at one place where we have also other patients with similar mutations, and we can observe, how the existing drugs behave. Because you can only see patterns if you look at many individuals at the same time. If you only look at your own child, you have so many drugs in parallel going on, so many interventions, it's hard to understand the cause and to effect relationship. But if you have many, many, many patients next to another, and it can be even a simple Excel table, you might see suddenly some patterns. And this is really important for existing drugs. So the next step is the future, right? So in the future is bright, but it comes also with risks. So you might have noticed that some people have a little bit a critical view on vaccination, right? And this is with something which is generally considered as safe. But think about a drug, which you would give to an individual, and it would perfectly, so you expect that you need to be in this green zone, and you give the drug, and from that on forward, the individual would be always in the green zone. So like gene editing or gene therapy or specific types of ASOs. So a one-time solution. This is what all this precision medicine is about, right? That is the best case scenario. So you, for example, you don't have enough gene, let's call it simple like that, and you give this drug, bam, you're in the green window, you have always your whole life enough gene. This is what we want, right? However, variant interpretation is important because imagine you think that individual has a so-called, here you would think it has a loss of function mutation, right? So you want to bring that back to, normal. But imagine that individual had only like a partial loss of function. So the CACNA1A encoded protein works a little bit. So works a little bit, not just, it's not completely diminished, but it works a little bit. At the end, clinically, you might not be able to see that really, especially if you have limited knowledge about the disease. And you would say, okay, is in line with a loss of function disease. And then you would apply this level of the dosage of a drug, and then you shoot above it. Because at the end, it was only a partial loss of function compared to a complete loss of function. And imagine you would do it completely wrong. You think it's a loss of function, but at the end, what you have is a gain of function, and you just accelerate this because you give more gene than to an already existing hyperacting gene. So, and there might be extreme scenarios. So long story short, is we need to have a good knowledge about these so-called relationships from gene to disease or from gene to function. And this is something which we can do already now and by just bringing a lot of information together. So, and this slide looks maybe frustrating, but I think it's important to recognize that a lot of decisions are made with incomplete knowledge. And one of the most successful stories, which is where this is done on a daily basis, is epilepsy surgery. So for individuals which have a specific focal epilepsy, where the epilepsy starts at a specific region in the brain, what people do is that they get a so-called surgery assessment. It's like a patient management conference, and they do an EEG and see if it comes from a specific region. They get an MRI and see if there's something abnormal in a specific region of the brain. And then there's a discussion about the clinical history of that individual patient, and then the patient gets surgery. So in a procedure where sometimes even almost like my fist, part of the brain gets cut out of a human. And the success rate is around about 70%. So it's not 100%. So every time this is, someone gets evaluated, a decision has to be made with, again, incomplete knowledge. It's really important. For these kind of things, you don't get to 100% knowledge. So you get really good hypothesis for like electrophysiological experiments or clinical representation. Someone looks really like a loss of function, or this variant looks in a HEC cell experiment really like loss of function. But at the end, we want to go to a place where we can bring a lot of independent level of evidences together to then come up with a confident decision. So this is this famous example where this elephant is described, and there are a couple of scientists, and everyone is blinded, and everyone just says, oh, that's like a piece of whatever leather. The other one says, oh, it's a piece of whatever, and then because they're all blind, right? So, but at the end, if they would open their eyes and they would work together, they would realize it's an elephant, and that's the same what we need to do for every variant. So we need to have, like people do for epilepsy surgery management conference, where there are different experts, like radiologists, epileptologists, EEG specialists, and who knows, so other physicians. We need to have like variant management conferences and bring together basic scientists and clinicians specialized on migraine, clinicians specialized on ataxias, and then these information needs to be brought together, and then an educated guess has to be made. And we are in a beautiful situation today that there is already a lot of information from other ion channels even, and other domains where we can bring, can make or accelerate information and can focus then with the foundation of filling the gaps which, where the information is yet not here. Okay, so this is the whole concept behind this, the portal which we have developed. So at the end, the idea is to really collect information, make it, curate it and organize it, and then move forward to from knowledge aggregation even to algorithms. Okay. So with this, I will start now to present the portal, which is a really big team effort from many scientists which are here in the room, and I will shout out them individually, and has been done by these people at that table at the corner there. Okay, so with that, I will start mirroring my screen and show briefly the portal. It will be up only for like a couple days because it's still really work in progress. We just started with the support of the foundation just this year, and we hope that we have a more advanced version which we feel very comfortable to keep online by the end of this year. Okay, so this is the so-called CACNA1A portal, and the whole idea is really to bring all information together that you don't need to have at every place in the world a team of 50 experts, right? So that you basically, the information is organized for you, that you and the wider community, that you can learn about the disease and even educate your caregivers or other individuals to learn about the disease. So this is work in progress. The idea is here to highlight the main publications which we as scientific community think are most relevant. And again, this is work in progress. Here are a few selected examples. And then we are trying to organize now the clinical representation of individuals with CACNA1A in a way that it's not fragmented across like thousands of publications. Instead, it's organized in one single database, which many people with different clinical and genetic training have looked at and agreed on that this is the clinical representation of a person with a specific mutation. And this is mainly led by the team at CHOP, together with the Boston Children's team and also Northwestern with Al. So, and here are different ways to look at this. So, for example, in total, 280 individuals have been identified with CACNA1A mutations based on databases and also from Columbia, from the Natural History Study, which hope everyone signed up yesterday night. And out of those 280 individuals, with 80 have been already looked at with more detailed clinical information, and the information has been standardized. And the standardization process is very important, that we can make it machine readable, that we can do computation with this information. And here's just some statistics. I don't want to go in much detail, but you can already see that we can now do comparisons between people with have a so-called missense variant, these changes of the mutation, compared to people who have so-called truncating variants, where they have a loss of function. And this is just the beginning where we can then do statistics and see differences. And this website is online for the next days, and you can play with this. So then next, we generated together with the foundation and this info video. You can and here you can also link to the foundation. So if someone finds the website, for example, through Google, they find also the foundation. And so this video is, for example, on YouTube. So if you, for example, search then for where are CACNA-related disorders, you find this video. And I will just briefly skim through the video. I will not go in much detail, so I will try to do this if the internet allows me. Yeah. So here we describe now what are the CACNA1A related disorders, and we describe what is a protein, what is an ion channel, what does that protein do in the body, what are the associated diseases, what is the role of these genetic variants, how do they cause different diseases, and what are the current recommendation and things to look out for. So we generated this video that you can share this with family members or even your physician and other individuals that and it's on YouTube, right? So this is now like as a resource for sharing the disease, and we tried really hard to communicate in a good way. And if you feel that this information is not well presented, just let us know, right? So this is work in progress, and we also really need to hear from you what information is the next most important one, right? So that's the whole idea behind this online resource. This resource is homework for everyone. So literally, with every time someone of you is playing with it and you struggle with certain things, this is good information for us to make it better, or even potentially to do it better with you. So this is then this educational part. Then we have this information where you can enter a mutation. You can submit this mutation. You get some informations about the which might be relevant for genetic counselors or biologists about the mutation or variant which has been submitted. You see then if this variant or mutation has been seen before in our database. So you can see, for example, that this variant has been seen in 3 different individuals. And this is, again, this clinical part is still really work in progress. And so you can learn about the other patients and their clinical representation more about this in the future. And you can also do some other explorations, which will go much into detail. So for the genetic counselors, we have also then information about how this variant relates to other mutations which we found in our patient cohort or in the general population. So this is a little bit more specific. And here we needed to, for the research part, so we want to also enable scientists to explore our data more. We developed also this interface where one can look now at the individual variants with bioinformatic tools. So sometimes this can really be useful for biologists to look where specific mutations are located. So as, for example, again with this example of the car, if you, for example, would have a mutation in the radio, who cares, right? You don't hear music while driving, but you can still drive. But if you would have a mutation in the car brakes, you probably don't want to drive. You can drive, but you probably don't want to, right? But if you have a mutation in the engine, your car might not drive at all. So, and that's why, and if you think about this as a car, you can look now where the patient mutations are located, and biologists can then observe and think about this if there are specific regions have specific function which can inform the role of that individual mutation. So we developed then this interface here where one can not only look at the individual CACNA1A patient variants which have been collected through the literature, but you can also look at mutations from different other sources. For example, control databases or variants which have been functionally tested. For example, the variants from Jen, she shared it yesterday with us. We added them also here. So, and you can, let's see if that works. So the internet connection is here not the greatest. Let's see. Okay. Okay, that only worked halfway. I'm not sure what's happening here. Anyways, so what you see here, for example, is the CACNA1A gene, and this is basically the first building block of the CACNA1A gene, and this is the last one. And this has like 2,500 letters which build this protein. And you can see now here where our patient variants are located. And you can see Yeah, it's actually not. Yeah, you can see, for example, where we have then also mutations which have been seen in related genes, like in other calcium channels or sodium channels. And you can see where there are CLINVA variants. And then you can explore the variants even further. And this is more useful for genetic counselors because we also have a lot of variants here from control databases like Nomad or UK Biobank. But yeah, I don't know. So I guess that's the internet connection because it worked there. But yeah. Anyways, I just move on. So we can organize the data here in different ways, and we can also look at specific regions of the website, of the protein if there are specific types of diseases more overrepresented, but that doesn't work now. So that's unfortunate. Okay, but the link is online, and you okay? Some things work now. Okay. So you can look now at the different regions of that gene and see if there are different disorders are overrepresented. And so especially for biologists and genetic counselors, this part should help them really interpreting the variants. And again, the website is online and you can just select it yourself. Yeah, so I think it might be, yeah. So it's this link. So it's CACNA1A-portal.broadinstitute.org. And we can also, I leave it like this, you can also send the link around later. It will be online for a couple of days, and then we pause it and work further on it. So, okay, let me just try one more time because the team worked really hard on this, and I just want to show because it's cool. Okay, but that thing should work at least offline. Yeah, so, and this work is, many people have worked on this over the last half year, and this includes many teams who work on clinical data, so they see patients and organize the clinical information and sort them and reevaluate them. Same as the molecular data, and you have already seen multiple speakers today, and you will also see Al presenting next. And of course, this was supported by the foundation and at the end by you. And we got a lot of support for the video as well for feedback. And I'm really annoyed that this is not working. Okay, no, but it works, but looks weird. Okay, so let's don't do this now. So one thing, what I want to show now more at the end of this presentation is, so this is work in progress, right? So, and I wish that has worked now because I could be a little bit more. Yeah, I guess it's the internet. Yeah, but. Yeah, but it's fine now. So, yeah. Yeah. Okay. So, anyways, so we are aggregating now for every variant functional data, which has been done, or we are generating more and more functional data. We're collecting clinical information from every patient, which has been observed so far. We're integrating more bioinformatic tools, which help the biologists to prioritize variants for molecular testing. And we are really motivated to integrate also molecular data like functional, like from mouse models and so on to integrate in this portal. So, and I really so hope that at the end that at the moment you need a lot of energy to make sense of an individual mutation and how that relates to the disease. But with this information integrated, you can just enter it and then get all the information what this room of scientists at the end would know, just prepared for you. And if you have this information, then you feel more confident in interacting with your clinical care team because you have independent information which has been curated from scientists all over the world. So what is the goal of this? So beside knowledge which has been aggregated. And this is now really important, and I think we are like maybe a year away. Okay. Okay, I will do this at the end. Okay, if I remember, summer 22. Okay, so data is good, if you can aggregate it and can leave individuals up to use it and connect the dots, but it's even better if you have so-called decision support tools. And I want to just give you a few examples about decision support tools which we could build based on other portals which we have worked on just in the last year. So, and I'm certain that we can develop something for CACNA1A in the near future if everyone adds more data. So that's the important thing. So here's now an example for the gene SCN1A, which is in the rare disease community relatively famous because it's one of the best studied neurodevelopmental disorders. And here we developed also for SCN1A, but also related SCN genes. It's like CACNA1A, they're called like SCN1A, SCN2A, SCN3A. We developed this portal where it has similar features like the CACNA1A portal, but it's a little bit more rich in data. So we have data from 4 different genes, which are very similar, and we have in total like 2,400 patients included here. We're including 800 functional tested variants and also a little bit more bioinformatic features. But it's not far away, and we can learn a lot from this. And this is also like a foundation-led project with many people involved. And also, again, it's like a huge community-driven project. So what can we do with this? And it's quite interesting, and I think this is where we need to go, my personal opinion, and we need to go also for CACNA1A. So we were able for the gene SCN1A to, let's see, where's my mouse? Okay. To identify it through like a global collaborative effort, okay, here, 1,000 individuals who either had the very mild epilepsy, so it's so-called GAPS+, or the very bad, catastrophic epilepsy, where also a significant amount of children will even die because of the disease. So in the mild disease, people can go to high school and live independent lives, have children, whereas the devastating one, the children will not live one day alone. They need always help. It's a very different, difference in disease, what we observe from many ion channels. And the variants are, so the mutations are in the same gene. The question is now, what is the difference, right? So, and when we had 1,000 patients, what we noticed is that when the seizure starts, it's very different in both groups. In the Dravet group, it starts around month 6, and it's very narrow, plus minus 2 months, for almost 90% of children with Dravet. So biology is pretty strong in Dravet. However, when you think about, when you look at the GAPS+ children, you see that the seizures start around about month 5, but they go up to 2.5 years. And in particular, Eduardo actually did this project. So when he looked at this, that was already very informative, and we thought, hey, maybe we can use this for develop an algorithm which, or a guideline which can help to distinguish the groups clinically, because you want to treat as early as possible, right? But then we also developed a genetic score, and we noticed that also the genetic score can separate both groups quite well. So, and then we asked the question, and that was the important one, if we would combine the genetic score with the seizure onset, would that help us separating both groups or predicting both groups? And it turns out that we could do this. And the interesting thing was that if we look in our own cohort of, we could basically, out of 100 times, it would be 89 times correct if we want to establish if the patient has the mild or the severe disease. And this is good, but if you look at on your own data, you might do things which you don't notice, which make your results better than they truly are. So to see if our algorithm, which is just the genetic score and the seizure onset, if they can really distinguish the mild from the devastating disease, we reached out to two collaborators, one group in Australia and one group in Belgium, and asked if they want to participate also in testing our algorithm. But this time we only got the mutations, but we didn't got the classification. So we did it blindedly. So we didn't know if our classification is correct or not. And what you see here is basically out of 100 times, we were 94 times correct in predicting the mild versus the severe disease. In the Australian cohort and in the Belgian cohort, we were 92 times out of 100 times correct. And this is just data which came apparent that some bioinformatic genetics magic score plus the seizure onset together allow this strong separation, and that has been blindedly validated twice. So this is something where I, as a scientist, feel very strong about using this as a second opinion. You have to, you know this yourself, right? Even in the largest academic centers, the experts, the global experts, they will not see more than 10, 15 patients in their lifetime with the same disease. But this thing is trained on 1,000 patients from experts which have curated that data. So using this as a second opinion, I feel very strong about. Of course, we have developed it, right? So, but anyways, so this has been published, and this is one example how if you aggregate data, you suddenly can do things which you cannot do if everything is all over the world. You can suddenly perform kind of statistical methods which are possible only with expert-curated and aggregated information. The second example, and this is unpublished, is from another ion channel disorder, the so-called GRIN genes. And here we developed also a portal, and the interesting thing is we have also the similar interface, what you couldn't see today, here. And when we looked at where the patient mutations were located, and we separated them once in those which are tested, like Jan Pan did for CACNA1A, but here Steve Charnelis did it for these GRIN genes. We looked at where are the loss-of-function mutations located on this channel, and where are these gain-of-function mutations located on this channel. And you see that the gain-of-function variants are here in the middle of this flower-looking thing, and the loss-of-function ones, they're in this upper part. You can, by eye, suddenly see that there is a big difference, and you see this only if you have numbers. So, and suddenly we saw this clear difference, and that motivated us to think about, hey, maybe we can develop an algorithm to separate the disease mechanism-based mutations. And this is very urgent, actually, because there is a company, so Therapeutics, which has a clinical trial out at the end of this year, but the drug only works for gain-of-function mutations. And this will happen to this community as well. There will be suddenly one drug faster than the other, and it will only work on a specific mechanism. So here, being in the position to say, okay, you should be ready, because, and the others, you should not participate because it might be dangerous for you if you would participate. We need to organize all our variants, or mutations, based on this kind of information. So the interesting thing was, based on the observation we could do, because with the data we had in the portal, we could then ask the question, is it because there are these so-called binding sites, which are known for biologists here in the upper part, where the loss-of-function variants are more close, and maybe there's in general a relationship with function to binding sites, because this type of ion channel is actually called so-called a ligand binding channel. So actually, the answer is already in the name of the channel, but no one realized that really. So it's stupid. Anyways, so yeah, really, that's like 20 years. The thing is called ligand binding, and so maybe it's important for function, right? So anyway, but I don't want to discredit my colleagues. That was somewhat known. No one put it in an algorithm. Okay, so so the interesting thing is, if you look now the distance to any of these ligands, so this is a measurement for distance, could be yards, meters, whatever. You see that the patient variants are closer to these glutamate binding sites compared to mutations which you find in people without neurodevelopmental disorders. You can see a striking difference. They're way more closer. And this is true also for these glycine binding sites. So if the mutations would be located here, the patient ones are closer to this area. And if you include this information into algorithms which say if a mutation is bad or good, that helps, this information adds to the algorithm that they almost get perfect. So with this simple information added to existing algorithms which say mutation is bad or good, we get almost perfect classification. So we can reduce the burden of VUSs significantly with a lot of confidence, and it's biologically interpretable because we suddenly understand also why it's bad, and because it's here, it's close to the glycine bindings and it's needed. Because if that thing cannot bind anymore, then this whole thing cannot act anymore. Imagine you would have like a computer without keyboard, or you have suddenly all your keyboard would be messed up. So, and the interesting thing was now, can we predict function, right? So the next question was, if we take, so we were really lucky that for the NMDA receptor, Steve Tranelius group, he had more than 400 patient variants and also some population variants functionally tested. So for example, Jen is at 40 to 50 at the moment, but here for this gene, we had 400 already. And so we could then ask the simple questions. Are, for example, loss of function variants to the glutamate binding sites, so basically to this one, or glycine, this is what we saw by eye, are they statistically closer than, for example, gain of function variants? And this is work was also Tobias did. And you can clearly see it by eye, that these loss of function variants at glycine and glutamate binding sites, they are way closer compared to the gain of function variants. And the other way around, if we look at this in this channel POR, at the magnesium binding site, you see that the gain of function variants are way closer than the loss of function variants. So, and with that thing, we can also develop then algorithms which predict, let's say, around about 80 times out of 100 times correct the function. So this is not perfect, but we have some rules now. We can say, hey, this is really close to the binding site, and we currently improving these algorithms because we can add more biological information to it from biophysics, and we are at the moment, I think, at 88% correctness out of 100%. So this is not, it's still work in progress, but the point I want to make is suddenly if you have a lot of information, you can use this information to make better guesses. So, and this is what the next phase, right? So we're doing not the portal only to have information lying there that people look at it. We want to use this to, de-risk information. And at the end, we don't want to be this blinded scientists, right? So which individually have an opinion. We want to bring that all together, that we see the elephant, and this kind is the different versions of CACNA1A disorders. And to do this, we really need to, aggregate all kinds of information. So we really need to get, clinical data. This is, for example, the longitudinal study. But at the end, we don't need to. We should do this not only based on parent-reported information. We need to get the hardcore biomarkers for all the MRIs, all the EEGs, all the other assessments, and see how they move across time in different individuals. We need to get the deep genetics, not just the panel, even more, to look at this more comprehensively. We need to get all the molecular data from each variant and organize it in a nice way that this information can talk to another, that we can really make the best educated guess based on available information for every variant. And when we have this, and then we have, as a foundation, then done our homework and are ready, right? For some variants, it will be more easy than for others, but at the end, with every information, we as a community move a step forward. And this is really then encouraging the scientists to generate data and share existing data. And this part, for example, is existing, right? This is just hidden in many patient records across hundreds of different hospitals, but it's there. We just need to get it. And this information needs to be organized in a way that it's comparable from one mutation to another. So again, this work, what I just presented and where we are moving towards, is a big team effort. And there are many individuals who, from the community, really led initiatives from the molecular to the clinical side, from the genetic interpretation side. And I think these examples, what we have observed for the other portals, which are more ahead, these disorders might have been studied maybe 10 years longer in more depth, but we have learned so much, and we are still learning from the other disorders. I think we are just maybe a half year behind. So, and in the space how we catch up, and I'm saying this again, I think this will be the most frequent neurodevelopmental disorder among ion channel disorders. I might not have a clue of anything, but I have a strong opinion on this. And I really have a strong opinion on this. It's just the energy we as a community spend getting the word out and facilitating, motivating others to look at CACNA1A and pushing really hard to take the information which is there and giving us that we can integrate it in this resource, and then we can develop algorithms or generate educative guesses. And with this, I want to really thank the Foundation for the opportunity to speak today, and also the table over there from our research group, including Eduardo, Arthur, Tobias, and Alina, and of course, Lea for guiding me for CACNA1A. And overall, just thank you very much for being here. And yeah, thank you.
Thank you, Dr. Lal. I think due to the sake, or for the sake of time here, so we don't run into lunch too much, and also we don't want to cut into Dr. George's talk too much either. I have 2 questions from parents on the virtual end, and then if anybody here in person has a question for Dr. Lal, we would encourage you to talk to him during lunch. So thank you for your understanding. Okay, there's a lot of really excited parents and people out there about this. So I just want to let you know the virtual feed is very excited. We had a parent ask, is there a way or a capability to print out a variant specific report that could be used for a school's IEP, like an individualized education plan or program?
So we have something like this in place for this Grin gene disorder group, and it's relatively, I think it's 3-4 months away. That we can generate a report if we think it based on guidelines, if the variant is pathogenic and if it has been reported before and in which type of version of CACNA1A disorder, and if it has been functionally tested in CACNA1A or a related gene. But this will probably, I don't understand how that could be helpful for the school.
Oh, right. So with the IEPs, I'm sure a lot of parents here have kids with those. Those are individualized education plans that are put into place based on any special needs or special support services that the child will need. So I think what would be useful is if they can print out my son has a variant XYZ in CACNA1A gene. Common symptoms or disorders include, speech or global developmental delay, cognitive impairment, things like that can help the school sort of direct the curriculum and get the students the support they need. Yeah. So I don't think that they would be interested too much in the molecular characterization of the variants, more of the variants and then correlated or linked phenotypes.
Yeah. So if the patterns are strong, so one could say other children which have a similar variant and CACNA1A have the following symptoms later in life or at that age range. And this is what in the needs which are likely to be expected. But we can do this descriptively, just statistics, but I think the portal will not be in the position to generate a certified report. I think that's with the individual caregiver needs to do this, but the information will be presented in a way that if you would go then to the portal and show this your caregiver and say, would you agree that, then I think the caregiver might be in the position to agree. And we have a larger team which might be able to help, communicating some things also in the clinical realm further.
Okay, thank you.
And just to add, to the extent that it's helpful that for the foundation to include its resources on the portal site similar to where you have the education materials. We could add links to brochures, so if people find this portal, or clinicians or educators find the portal before they find our foundation, that all the education materials would be listed there. So we can work with you guys on that.
Okay, great. And then the last question here, there was a parent who asked, is when you're thinking about precision medicine, and maybe you touched on it earlier, is the targeting of gain of function or loss of function as these separate classifications complicated by a mixed function, right? Like, how do you handle that then?
So this is a question which the next speaker can answer better than me. The only thing what I would like to add is, this so-called gain versus loss of function is a simplification, just basically a slang, how people communicate variance at the moment. And at the end, sooner or later, we will move probably towards change of function version 1, change of function version 2, version 3. And the individual therapies are probably designed to tackle one or more of this, and the experts of each therapy might be able to comment which one works best.
All right. Thank you again to Dr. Law. And